synthetic organic chemist with a background in prebiotic chemistry. And this particular session is very near and dear to my heart because it's close to what I studied in my PhD, which is prebiotic nucleotide synthesis. Now, prebiotic nucleotide synthesis is a central and perennial conundrum in the study of the origin of life. I don't think I have to belabor its importance to this audience, but just to give you a taste of why it's so difficult. If you observe the structure of a nucleoside, you'll find that it has a heterocyclic base, that's the recognition unit, ribose or deoxyribose sugar, the trifunctional connector, and then an inorganic phosphate, which is linked by a monoester in the case of the monomers, or a diester in the case of the polymer. Now, if you make those traditional retrosynthetic disconnections like an organic chemist would, to synthesize that molecule in a prebiotically plausible manner is quite challenging. It turns out that life does synthesize it in that manner, but it requires activation chemistry to do so. And to mimic that in a prebiotically plausible manner is quite challenging. I believe so. A number of people have taken swings at this problem. I think Leslie Orgel was the first to attempt it in earnest. And the conditions that he found were quite harsh and gave quite low yields of these canonical nucleotides. But since then, a lot of progress has been made. And I'm very excited for our two speakers, Nick Green and Yuta Hirakawa, to present their research, which offers some solutions to these problems. So our first speaker will be Nick Green. Now, I know of Nick from his work with John Sutherland, but he is now starting his independent career as a lecturer in the chemistry department at the University of Otago in England. He completed his PhD in organic synthesis at the Australian National University, and he then continued with organic synthesis and methodology development at the University of Edinburgh, and then joined the Laboratory of Molecular Biology at Cambridge, working on origins of life, as I mentioned before. He started his independent career in 2022, and his group studies organic and biological chemistry related to new methods and structures and the origins of biological systems. So, Nick, thank you very much for joining us today, and take it away.
Thanks very much, David. Can everyone hear me okay? Cool, I'll just share my screen. We can all see that?
Looks great.
Excellent. Yeah, well, thanks very much, David. It's great to be here. This is my first PCE 3 seminar meeting, so I'm looking forward to attending more of these in the future. And yeah, I'm looking forward to some interesting discussion after that really nice introduction from you, David, on the subject of prebiotic nucleotide synthesis. So as David mentioned, I've just moved to Otago. I moved to Dunedin, the University of Otago in Dunedin, New Zealand, about a year ago now, and I started my own group. But as we have got some interesting little bits and pieces of results, but it's only been a year. So as this is my first time at this seminar, I think it's a good idea to present some of the sort of big story that I was involved in when I was working with John Sutherland in his lab in Cambridge. And that's this work on prebiotic photochemical link between RNA and DNA. Cool. So I want to start off the talk by just asking this sort of grounding question: why does DNA exist? And of course, there are lots of different sort of prevailing theories, but I think possibly a simple and common answer is that DNA is really good at its job. So DNA, of course, forms part of the central dogma of biology, where we have these three classes of biomolecule: DNA, RNA, and proteins. And essentially, it's the interaction between these three classes of biomolecule that dictates most of the biology that we know. So why is DNA good at its job? Well, it comes down to a number of different, sometimes complex reasons, but if we contrast the properties and behaviors of RNA versus DNA, we can see that DNA overall, based on a number of sort of structural and stability factors, DNA tends to be the better molecule for stably storing and replicating information. And so it fits beautifully into that first part of the central dogma. However, as we know in life, and of course it's true in prebiotic chemistry as well, just because an outstanding solution to a problem exists, that by no means that it will actually be found. So what I'm going to talk about today is perhaps how biology was able to stumble upon this relationship and this sort of duality of nucleic acids in the central dogma of biology. So just before we get into that, a little bit about the sort of prevailing theory of the origin of life. There are many different theories to the origin of life, and different ones are gaining momentum as well. But by far and away, the most sort of common idea is the centrality of RNA at the center of the origin of life. So people have different ideas about what exactly an RNA world would have looked like, but in broad strokes, we can think about the formation of the Earth and then geochemical processes taking place on that Earth early in the history. That will give rise to some sort of chemical system where the sort of key moment in the emergence of biology is when RNA is able to take over. And I'll get to why we think RNA is able to act as a biological system in just a moment. And able to self-replicate and essentially carry out all those functions in the central dogma. But the prevailing theory at the moment, or certainly recently, has been that this key early stage was almost exclusively based on RNA as the nucleic acid, as the genetic system in that first form of life. And that DNA was a subsequent biochemical invention of this initial early life form. So to simplify that a little bit, look at it slightly differently in the bottom of this slide here, we have our prebiotic chemistry giving rise to RNA. In the RNA world, there's certainly lots of room for other molecules, peptides and lipids, sugars, other good examples of important biomolecules. And that gives rise to this first sort of idea of protobiology, where RNA is playing the dominant role. And it's then the status quo of thinking is that this protobiology has access to rudimentary biosynthesis. And one of the key innovations that this biosynthesis is able to access was the biochemical synthesis of DNA. And then in the way that evolution works, biology was able to exploit this biosynthesis of DNA and exploit the superior information carrying properties of DNA to then go on and diversify as we know it today. But the research I'll show you today is going to sort of get to that in a second. So just a couple of pieces of evidence as to why people think RNA is such a central molecule in that early stage of the origin of life. Basically, it comes down to the idea that RNA is thought to be able to carry out all the functions of the central dogma by itself. So we know that RNA can store information just reasonably competently, almost as well as DNA. We also know from fairly recent studies in the last few decades that RNA is also a competent functional catalytic molecule. And so therefore it's able to carry out many of the roles that we envision proteins possibly would have had to carry out. So therefore you can see that RNA can take up the role of all three biomolecules in that central dogma. A couple of other pieces of circumstantial evidence are the fact that RNA is ubiquitous in biochemical processes and metabolic processes in the form of cofactors. And it's really only in the last, I would say, 20 years or so or less that, as David mentioned, RNA, nucleotides in general, they're complex molecules. They're difficult to synthesize. They have lots of inherent complexity. But it's only really in the last 2 decades or so that people have, at least in broad strokes, defined sequences that appear to be prebiotically plausible. And, you know, there's certainly some fine tuning about the details of the scenarios that are invoked for those syntheses that needs to be worked on. But yeah, I would say in the last couple of decades, people are beginning to accept that this sort of complexity can be accessed via spontaneous geochemical non-enzymatic processes. One other aspect of the relationship between RNA and DNA that sort of adds to this idea that DNA is a late descendant or a biochemical descendant of RNA is the modern biosynthesis of DNA. And so until humans were able to chemically synthesize DNA in the laboratory, all of the DNA on Earth went via the intermediacy of an RNA molecule and was biosynthesized via this enzyme ribonucleotide reductase, which converts this 2' hydroxyl group of an RNA molecule into the 2' hydrogen atom that we see on a DNA molecule. So of course, even if we accept this idea of the centrality of RNA, and we accept much of the really pioneering work that's happened in the last 70 years really in prebiotic chemistry, of course, there's still this defining challenge that we need to find what the missing pieces of the puzzle are. So, we certainly haven't been able to solve the origin of life question. We've seen bits and pieces of the key molecules maybe emerging as prebiotically plausible, but we have to keep asking what other molecules would have coexisted in that primordial soup. That's a term that I'm going to mention a lot today, and flesh out the potential roles for those other molecules that would have existed alongside RNA, and look at the interactions between all of these molecules that could eventually give rise to a protocell. So just coming back to this timeline, the research I'm going to show you today is going to suggest that perhaps rather than being this biological invention, an invention of biosynthesis that biology was able to exploit, perhaps DNA, like RNA, is an early biological appropriation because it was a geochemically plausible molecule that existed alongside RNA before biology actually got going. Okay, so as David mentioned, I did my postdoc with John Sutherland. So the work I'm going to be presenting today was part of this wonderful team in John's lab. This photo is a little bit old now, but yeah, there we are. And also with our collaborators, Ram Krishnamurthy and Clementine over at Scripps in California, and Rafael Zablo, who was in Edinburgh at the time, but he's now moved on to Poland. So as you can see, I'm going to present a couple of chapters in this work. Most of it's been published, but of course, it's always nice to see someone talk through it. And yeah, hopefully it'll provoke some interesting discussion on nucleotide synthesis. So before we get into the new chemistry, when we talk about this primordial soup, of course, it's not just a vague concept. I know it's nice to think of a vague bubbling broth or something on a primordial landscape. But of course, there's loads of research that's been done into what the constituents of this primordial soup could be. And this is a nice little summary of the work in John Sutherland's group that was existing when I joined the group. And we don't need to think about the specifics here. So, it's enough to think of this as a primordial soup that's composed of actual real molecules and relationships between molecules via synthetic chemistry. And of course, it's important to point out that, from simple molecules like hydrogen cyanide, for example, in relatively short order, you can move across these types of networks and relationships to molecules as complex as nucleotides, for example, lipids up here, and of course, amino acids are scattered all throughout this network as well. One thing I do want to draw your attention to in that sort of network of reactions, or in that primordial soup, is the one carbon homologation reaction from HCN. So if we take a nitrile and we do a 2-electron reduction of that nitrile, that gives us an imine, which is of course in hydrolytic equilibrium with an aldehyde. And either of these two species can undergo addition from a molecule of HCN. And what that does is it forms a new carbon-carbon bond, and it also reforms the nitrile group. So you can imagine this substrate at the end of this sequence now being a substrate for the beginning. And in this way, you can homologatively form carbon-carbon bonds one at a time. And this is really important. I'll come back to in a second, the synthesis of the carbon-carbon bonds in biomolecules, we think that would have happened prebiotically. And so the source of electrons, there are a few different possible sources of electrons or a reducing agent that we can invoke in a prebiotic scenario. But an excellent one is this volcanic gas, hydrogen sulfide. When it dissolves in water, it forms an anion, hydrosulfide as shown here. And anions like hydrosulfide, and there are other anions as well, when they interact with a photon of UV light, they undergo this process called photodetachment. That generates a solvated electron, as well as this oxidizing radical here. But this solvated electron is the reducing agent in this chemistry up the top here. So if we come back to that prebiotic network, we can see this homologative sequence takes us from cyanide up to this 2-carbon sugar building block here, and then to this 3-carbon sugar building block here. And it's through the unification of these two building blocks along with a nitrogen building block too, that we get to most of the framework of a nucleotide. And so this was work that was existing in John's group. And what Jimmy had shown is that if you take this molecule RAO and you react it with cyanoacetylene, then you do this key reaction where you do a thiolysis with hydrosulfide, and that thiolyzes this CO bond here, and it generates this thiocytidine molecule, but you can see it's got the wrong stereochemistry here. It's got the alpha stereochemistry at the 1' position, but it's having this sulfur atom in place that allows a photoepimerization to correct that stereochemistry. And from there, all that was needed to be done is hydrolyze this sulfur bond here, this carbon sulfur bond, to form the 2 canonical nucleosides of pyrimidine nucleosides of RNA. So that's uridine and cytidine as shown over here. But what seemed to be a byproduct in this chemistry is this molecule here, 2-thiouridine. And so this comes about from hydrolysis of this CN bond, but not hydrolysis of the carbon sulfur bond. And so we sort of come to this idea that if you discover chemistry that you think really is prebiotically relevant, perhaps there's no such thing as a byproduct. Perhaps everything is useful. And so if you consider further this 2-thiouridine molecule, even though it's not a canonical pyrimidine, it is actually commonly present in biology today as we know it. It's a commonly detected tRNA modification, for example. So it pops up in RNA here and there. And also it's been shown to be important in the non-enzymatic RNA replication chemistry that Jack Shostak's lab has been doing for some time now. And so therefore we had this idea, well, okay, well, 2-thiouridine, it's not a byproduct of this chemistry. It's actually something important. But to feed into this chemistry from the Shostak lab, you'll see that they use the activated 5' phosphates. They use the imidazolides of the 5' nucleotide of 2-thiouridine. And the byproduct from the chemistry that was existing in the Sutherland group was the nucleoside, the unphosphorylated nucleoside. So we initiated this research program with a really simple question. We just asked, are we able to phosphorylate 2-thiouridine and therefore give a prebiotic provenance to this chemistry that was happening in the Shostak group? So to cut a somewhat moderately long story short, we did a bunch of reactions and we found that under what we think of as kinetic phosphorylation conditions, so this is a mixture of inorganic phosphate and urea in formamide as a solvent. If we heat this up, then we can get what we think of as the kinetic phosphorylation product, and that's the 5' phosphate. That's exactly what we wanted. Get that in about a 20% yield, recover a bunch of starting material as well. So in prebiotic chemistry, that's a pretty good result. We're happy with that. That's the activate, well, that's the 5' nucleotide, so that feeds into Jack Shostak's chemistry. So that was a successful result. However, we were noticing this wasn't the only product under many of the conditions that we tried. And we found particularly under more forcing phosphorylation conditions. So for example, if we heated it up in the solid state with just inorganic phosphate and urea, then eventually we detected these compounds as shown down here. So these are thio and hydrouridines in various states of phosphorylation. And so we realized what was probably happening was that we were getting the thermodynamic phosphorylation product, and that's the 2', 3' cyclophosphate rather than the 5' phosphate. And that this cyclophosphate's got an activated 2' position that can undergo this 5-exotet cyclization reaction with this nucleophilic sulfur atom to generate this thio and hydrouridine as shown here. As you can see, we got about a 50% yield of this molecule in its various states of phosphorylation. So again, initially we're a little bit disappointed. Okay, well, much of our material is going towards this byproduct. But of course, thinking that we're in a useful area of prebiotic chemistry, we had to use that different attitude where perhaps there's no such thing as a byproduct. Perhaps this is a useful molecule as well. And so we realized that having a CS bond in the 2' position makes that 2' position much easier to reduce than having the typical 2' CO bond where you have a hydroxyl group in RNA. So we thought perhaps this is an opportunity to convert RNA derivatives into deoxyRNA derivatives or DNA. So what we needed to do is we needed to reduce this carbon-sulfur bond, looking at this from a synthetic chemistry or retrosynthesis point of view. And we'd also need to hydrolyze this carbon-sulfur bond over here. And that would give us to this canonical DNA molecule, so this is deoxyuridine as shown on the right. So how will we go about doing this? Well, the reason I showed you this 1 carbon homologation chemistry before is because we want to see consistency in the geochemical scenarios that we're invoking for the synthesis of all of these molecules. Okay, we can't have different types of conditions for different parts of that network that I showed you. So if we want to reduce a CS bond, why don't we use the same reducing conditions that we used to reduce these nitriles earlier? And so the source of reduction power would be these hydrated electrons coming from the photodetachment of electrons from anions. So we went ahead and took our molecule and treated it pH 7 sodium hydrosulfide in the presence of UV light at 250 nanometers. And again, to cut a complex analysis story a little bit short, we got a nice yield, a 33% yield of a deoxy derivative. So you can see that the sulfur is still in place on the nuclear base. So this is deoxy 2-thiouridine. And so we think the mechanism for this process is that we go via that solvated electron that's coming from photodetachment of hydrosulfide. That generates this radical anion, which can undergo CS bond scission to generate a carbon-centered radical. That carbon-centered radical can pick up a hydrogen atom from a suitable reducing agent. And of course, when we have hydrogen sulfide in solution, that's a competent hydrogen atom donor. We also saw a bit of decomposition, a bit of a byproduct that we couldn't quite characterize. It was a little bit unstable, but we think it was probably recombination of this carbon-centered radical with this oxidizing radical that's a byproduct of the solvated electron formation. And that would generate this 2' mercapto molecule here. But again, we weren't able to isolate that molecule. So we had here 2' deoxythiouridine, and of course, that was a great result. We were able to reduce the 2' CS bond. But even though the RNA molecule of thiouridine, so thiouridine itself is in modern biology, the deoxy derivative isn't. So this isn't a molecule that's present in biology. So we were still very keen to see if we could hydrolyze this carbon-sulfur bond and to see if we could phosphorylate this molecule and therefore make it a part of nucleic acid polymers. However, anything we tried in this respect didn't really work at all. So almost anything we did led to the decomposition of this molecule. It wasn't a messy decomposition though, so it was a rather clean hydrolysis of this CN bond here. And so, under a variety of mild conditions, we basically saw this fall apart where we lose this labile glycosyl CN bond because this 2-thiouryl is quite a competent leaving group. And again, initially we were disappointed, but then we realized, well, actually, this is quite a clean synthesis of deoxyribose. And perhaps that deoxyribose could feed into some of the prebiotic chemistry or the proto-metabolic networks that were emerging at that time, and perhaps be a useful building block in the synthesis of DNA. And also we thought, well, chemically, we could exploit the weakness of this CN bond as well. And so the way we went about that was by thinking about trying a transglycosylation with a canonical nuclear base, because of course this 2-thiouryl is not a canonical DNA nuclear base. So the way we did that was we basically replaced the water, these aqueous conditions, with the dry state, having adenine as our nucleophile. And indeed, rather than water displacing this CN bond here, we were able to generate both products of substitution that you would expect from adenine, in a similar manner actually to the work that Orgel was doing, that David mentioned at the beginning of the session. And we found, similarly to Orgel, very, very low yields and poor selectivity. So we got a 4% yield of the correct isomer of beta-deoxyadenosine. So that was fantastic. That was the first prebiotic synthesis of a DNA derivative. But at the same time, we were getting more of the wrong isomer. And really, these yields weren't anything to be too happy about. So a quick summary of where we got to at this time. We had this existing synthesis of the RNA pyrimidines that was in blue. And as I said, this got to the RNA pyrimidines, which are shown over here, but one of the byproducts was this molecule in the bottom right, which is 2-thiouridine. And we showed via this phosphorylation reaction, which generates this thioanhydrouridine molecule, we're able to then photo-reduce that CS bond to generate a variety of DNA building blocks, but no canonical DNA nucleosides yet. So we went back to the drawing board and we thought, well, the way we do this reaction is coming back to the sort of mechanism of the formation of this thioanhydrouridine is we activate the 2' position towards this intramolecular nucleophile, the sulfur atom of the 2-thiouryl nuclear base. And so we thought, well, there are other intermediates in the already existing pyrimidine synthesis, this one, for example, here, that have an activated 2' position. So perhaps we could intercept this in an intermolecular sense and therefore end up with a more direct route towards this, because this is an earlier intermediate in that pyrimidine synthesis. So we tried out this reaction with thiouracil in an intermolecular sense and our new electrophile over here. And this was a successful reaction. So in almost in quantitative yield, we got a one-to-one mixture of the two regioisomers that you would expect from this reaction. So those two regioisomers, first you have nucleophilic displacement of the CO bond to generate this intermediate here. And then you can see that there are two nitrogen atoms that have this 1,3 disposition towards sulfur. So they can close up this 5-membered ring, either of those two nitrogen atoms, to generate these two regioisomers here. So this regioisomer on the left is what we think of as the correct regioisomer. If you go ahead and reduce this carbon-sulfur bond, then that gives you the canonical regiochemistry of DNA. And this regioisomer on the right, that's the incorrect isomer. If you go ahead and reduce that isomer, that CS bond, then you get the wrong regiochemistry for the resulting nucleoside. So that was a nice, more direct route, but what really got us excited about that result was the fact that if we're able to do it with thiouracil, then perhaps rather than looking at chemistry that was unique to the 2-thiouridine that we were able to synthesize, to the N-hydrouridine that came from the 2-thiouridine, perhaps we could make this more general with respect to other nucleosides. And so perhaps we could look at installing different nucleobases via this intermolecular displacement reaction, this tethered glycosylation or 1,2 glycosylation. And so we thought about installing a purine nucleobase. And so we'd need this nucleophilic sulfur in position, so an 8-mercaptopurine. And we thought about it doing an almost identical reaction, just displacing this CO bond, generating that similar intermediate that I would have shown you. And that, if we can get this chemistry to go, that would give this thioanhydroadenosine type molecule. And if we go ahead and do the reductions and the hydrolysis that we were working on before, that would link to the canonical molecule of DNA, deoxyadenosine. So how did that go? That worked out quite nicely as well. So again, we had a high yielding 1,2 glycosylation reaction with 8-mercaptoadenine. This time we didn't get a 1-to-1 mixture of the two regioisomers. We actually got a mixture that was enriched in the wrong isomer. So remember, there's two possible nitrogens that can close that 5-membered ring in the product. The N9 is the correct one, the one that we want. So if we then go and reduce this bond, we'll go ahead and get the canonical molecule of deoxyadenosine. The N7 isomer is the incorrect isomer. So we had a bit of a problem with selectivity here. You can see we got a fair bit more of the incorrect isomer, but I'll come back to how we solve that in just a moment. First, I just want to demonstrate that we didn't just pluck this 8-mercaptoadenine out of thin air. So we showed that the adenine itself, which is of course a cornerstone of prebiotic chemistry, the pentamer of hydrogen cyanide, it undergoes hydrolysis to this molecule triaminopyrimidine here. And this triaminopyrimidine, even though it's formed in relatively low yield from this hydrolysis, it undergoes a quantitative ring closing reaction with either thiourea or ammonium thiocyanate in the dry state to generate 8-mercaptoadenine, which is rock stable and precipitates out of this solution here. So we think there's a reasonable way of accumulating these molecules. So undeterred by our lack of selectivity in the tethered glycosylation, we went ahead and tried our photoreduction on the mixture. So remember, in this mixture, we've got more of this incorrect N7 isomer than we do of the correct N9 isomer. So we used our prototypical anion, our reducing agent hydrosulfide, UV light, and we found something interesting here. So these compounds on the left are both N9 isomers. The compounds on the right are both N7 isomers. The compounds on the top are the fully reduced DNA derivatives, the DNA nucleosides. The compounds on the bottom, they've had the 2' CS bond reduced, but they've still got that 8-mercapto group in place on the nucleobase. But it turns out that if you just continue irradiating these, then that 8-mercapto group is photolysed to give the correct nucleobase. But what was interesting was that even though we started with a mixture enriched in the incorrect isomer, we got a higher yield of these two N9 compounds, this left-hand column. So we got about a 55% yield of these two compounds, and we only got half that for the N7 compound. So straight away, we'd seen going from a wrong ratio to an enrichment towards the correct ratio. And we rationalized that by the hydrolytic stabilities of these two products. We found that the incorrect isomer is about 70 times more labile to hydrolysis. So we thought that it was probably being converted from its N7 starting material, but just falling apart quite quickly under the reaction conditions. I mentioned that there are other volcanic outgases that can be used as an anion for a reducing source in a photodetachment process. And bisulfite is another one of these. Bisulfite forms when SO2 is dissolved in water, and SO2 is one of the major components of volcanic outgassing. So we also tried the same photoreduction conditions, but rather than hydrosulfide, with bisulfite as our reducing anion. And what we saw there was an even more remarkable consequence or outcome in selectivity. So we took this mixture, N9 and N7, and at the end of this reaction, the only material that we observed was N9 configured. And so what had happened was, in the reaction, all of this N7 material had been completely destroyed. And this was a sort of destruction. It wasn't just a clean falling apart of the nuclear base. All of that material was essentially chewed up into material that wasn't observable, at least by NMR. So we got a really quite high yield. About 75% of this material was cleanly converted into these two compounds here. Essentially, all of that gets converted to the canonical N9 regioisomer of deoxyadenosine. So that was a really great result. So we'd shown that even though our glycosylation reaction had limited regio selectivity, the photoreduction corrected that limited regio selectivity and gave us complete selectivity for the canonical isomer, the canonical stereo, regio, and chemical isomer of deoxyadenosine. So the mechanism for this photoreduction, I won't dwell on it. I know we're running a little bit short on time. So basically, we just think that the photoreduction proceeds differently for the two regioisomers, obviously. And so the first engagement with a reducing species for the correct N9 isomer basically results in very clean, very fast scission of that CS bond to form a carbon-centered radical. Whereas the N7 isomer, it doesn't cleave that CS bond, and it instead forms a radical anion, which then undergoes various forms of decomposition. Hydrogen sulfide, the reason it doesn't undergo that complete decomposition for the N7 isomer is because the hydrosulfide actually forms this encounter complex, which pre-organizes the reduction to occur with scission of that CS bond. And so that's why you see a different outcome in the reactions between bisulfite or hydrogen sulfide as the reducing agent. Okay, so where had that got us to? Well, we'd shown that from an existing intermediate in the synthesis of the pyrimidine RNA nucleosides, we're able to divert that molecule and take an intermediate in that synthesis, react it with this glycosyl acceptor over here, and show that we could do this really clean tethered glycosylation reaction, which subsequently followed by a prebiotic photoreduction would give us the canonical purine isomers of DNA. So I didn't mention, but it's really easy to convert this deoxyadenosine molecule into deoxyinosine at pH 4 in the presence of nitrous acid. And so if you look subtly here, that there was a pre-existing synthesis of the ribopyrimidines, but of course, there was a big gap there. We didn't have a synthesis of the ribopurines. And we still don't have a synthesis of the ribopurines, although there is some later work that I won't have a chance to present today where we did actually manage to make them. But this clean synthesis that's really strongly related to this work here shows that we have a sort of complete alphabet, at least of nuclear bases, but only half and half with the ribo and deoxyribo sugar chemistry. And so that raises this question. Of course, we weren't the first to raise this question. This had been discussed in the literature before, but perhaps this was a prebiotic evidence or a prebiotic basis based on synthesis for a hybrid genetic alphabet at the beginning of life. So we have the ribopyrimidines, and flowing on from that synthesis, we have the deoxyribopurines. So that brings to mind this thought that perhaps one of the reasons we have these two systems of nucleic acids is because the first system was a hybrid of those two systems by necessity, because biology was only able to use these molecules that were in its environment because of prebiotic synthesis. And then once biology got developed and was able to access different types of biosynthesis, it was able to diverge into specializations for these two molecules into the roles that they play in the central dogma of biology today. So just to recap, again, the change we've made here is rather than thinking of DNA as a biological appropriation, as a biological invention that comes later, we're suggesting that DNA could be a biological appropriation that DNA is equally plausible to RNA, and therefore, rather than focusing on an RNA world, when we try to look at the interactions of molecules in this primordial soup and look for the emergence of biological properties, perhaps we should be factoring DNA into those discussions and experiments as well. Okay, so I'll finish my talk there. As I mentioned, I started my group here in Dunedin about a year ago. So of course, it's great to be able to present some work to a new audience here. And so if anyone knows of anyone or themselves is interested in pursuing some chemistry in a beautiful scenic part of New Zealand with our beautiful university here, then please just go ahead and get in touch. Thanks very much, guys.
Nick, thank you very much for that wonderful talk. I forgot to mention in the beginning that we'd like to hold all of the questions until both presenters have given their talks. So with that, we'll go right into the next talk. Our next presenter is Yuta Hirakawa, who is currently a PhD student in the Department of Earth Sciences at Tohoku University. He also obtained his bachelor's and master's degrees. His work has been recognized by the Geochemical Society of Japan several times. And he's going to discuss prebiotic phosphorylation, which I think is a nice flow from the previous talk where Nick touched on it. So Yuta, please take it away.
Yes, can you see my slides?
Yes.
Okay, thank you very much. Hello, everyone. Good morning, everyone. I'm Yuta Hirakawa, PhD student at Tohoku University in Japan. And thank you for inviting me to such a great seminar today. And today's presentation, I talk about the Borate-guided sugar phosphorylation for prebiotic nucleotide synthesis and the origin of life. And my research field is prebiotic chemistry. And my research interest is the origin of RNA and nucleotide. So, my first slide is almost same as what the first speaker Nixon said. The current life uses two types of nucleic acid and DNA and RNA. And DNA holds the information, and RNA copies this information and catalyze the reaction to make protein. And protein works as biocatalyst in our body to make DNA and RNA and protein cells. And of these three molecules, RNA can hold the information and catalyze some chemical reaction like protein. So many researchers consider that the primordial life used RNA to duplicate themselves. And this theory is called RNA world hypothesis. And based on this hypothesis, the formation of RNA is one of the essential steps for the origin of life. And RNA is a polymer of ribonucleotides. So in order to make RNA, it is necessary to synthesize nucleotide first. Nucleotides consists of three components: ribose, a kind of sugars, nucleobases, a kind of nitrogen heterocyclic molecules, and phosphate. From this slide, I introduce previous research on prebiotic nucleotide synthesis. The first prebiotic nucleotide synthesis was reported by Orgel group. which is a direct condensation of nucleotide component, ribose, nucleobase, and phosphate. They show that nucleoside synthesis from the direct condensation of ribose and nucleobase and the following phosphorylation. The challenge of this nucleoside synthesis is low yield. And no one knows how these molecules were prepared on the prebiotic earth. On the other hand, nucleoside phosphorylation to nucleotide is relatively simple. The reaction is high yield and one-pot reaction. So many researchers have focused on the formation of nucleoside synthesis compared to nucleoside phosphorylation. Recently, Thomas Carrel group have proposed a new reaction to form nucleoside from ribose, and they reacted nucleobase precursors with ribose, and they converted this intermediate to canonical nucleoside. The phosphorylation part is almost same as Auger method. This reaction proceeded with high yield due to the high reactivity of these precursors. But this reaction needs multiple step reactions, contrast to the one-pot reaction, one-pot nucleoside phosphorylation. This slide shows the most famous, probably the most famous nucleotide synthesis reaction proposed by Sutherland group. They reacted simple but very reactive molecules step by step and synthesized the nucleoside precursors. And then the precursor was phosphorylated to the nucleotide. Also in this method, nucleoside synthesis reaction was very high yield, but needs multiple step reaction, and phosphorylation was almost same as Auger method. So the common point of this previous research is the reaction order. The nucleoside formation first and following phosphorylation. Many researchers have proposed nucleoside synthesis reaction. On the other hand, phosphorylation has been already established. In other words, nucleoside synthesis is difficult, and phosphorylation is relatively simple compared to nucleoside synthesis. And please imagine the prebiotic earth. There's no chemist or scientist to manipulate or control the reaction. And there must have been primordial soup that contained various nucleotide precursors or organic molecules. So it is difficult to assume that the step-by-step reactions like nucleoside synthesis proposed by Carrel group or Sutherland group is proceeded rapidly on the prebiotic earth. Rather, simple reactions like phosphorylation should have occurred first. So we assume that phosphorylation occurred first, and nucleobase addition or nucleobase construction occurred later, which is different from the traditional pathway. And some previous research has reported the nucleotide formation from phosphorylated sugars, but no one reported the ribose phosphorylation, ribose phosphate formation from ribose and phosphate under the prebiotically plausible condition. So this motivated us to investigate the formation of ribose 5' phosphate. Then why is it difficult to form ribose phosphate? There are two reasons: stability and selectivity. First, ribose is very unstable molecules under the heat condition. Phosphorylation generally needs heat and dry condition, but ribose easily decompose or reacting each other in such condition and turn to brown tau. Second, it's difficult to phosphorylate the right position of ribose. The previous research tried the ribose phosphorylation, but only ribose 1' phosphate was formed, not 5' phosphate. For the chemical evolution for nucleotide and RNA, we need right structure molecule, and 5' hydroxy of ribose need to react with phosphate. These two issues make it difficult to phosphorylate the ribose. To solve these problems, what we focused on is borate. The borate will have existed in the evaporative environment on the areas, and borate can stabilize sugars by forming this sugar borate complex, and borate can keep the ribose as cyclic form, and cyclic form ribose is very stable, so borate can stabilize ribose. So the borate-rich environment can contribute to the accumulation of ribose on the areas. And borate also contribute to the regioselective phosphorylation. This research shows that nucleotide regioselectively phosphorylated at 5' position because borate can combines with this position, these 2' and 3' hydroxyl. So we consider that we can apply these borate abilities to ribose phosphorylation, and the objective of this research is to investigate the effects of borate and borate-rich environment on the phosphorylation of ribose to find a new route, another pathway for nucleotide. Okay, let's move on to the experimental method. Here is the method. For the ribose phosphorylation, we perform the thermal evaporation experiment. The experimental procedure is pretty simple. First, we prepare the aqueous solution containing ribose, disodium monophosphate, boric acid, and urea as a phosphorylation catalyst. The solution was heated for 24 hours at 80 degrees Celsius with the lids open. And after the experiment, through some pre-analysis process, we analyzed the sample by HPLC-MS. Here is the result of the experiment. The upper figure is the standard of ribose phosphate, and lower figure is the experimental product. The x-axis is the retention time, and the same retention time peaks means the same product, and y-axis is the intensity. The black line shows the ribose 5' phosphate. This is the target material. And the blue line is the ribose 2' phosphate, and yellow line is ribose 3' phosphate. These data show that ribose 5' phosphate was selectively formed in the experimental product, and this fragment pattern spectra also indicate this peak is ribose 5' phosphate. And we calculated the yield based on the peak area, and it was 22%. This yield is significantly high as a prebiotic reaction. Here is the result in the absence of borate. We detected small peaks that has same retention time and same fragment pattern spectra as the standard, but the yield was only 4%. This result indicates that ribose could have been phosphorylated under the prebiotically possible condition, and borate can increase the yield of ribose 5' phosphate. And let me summarize the results so far. In the traditional pathway, many researchers have focused on the nucleotide synthesis and following phosphorylation. But this study shows that ribose phosphate can be formed, and this alternative pathway, this another pathway was possible as a nucleotide synthesis on the prebiotic earth. Oh, and the next question is why ribose was used in the canonical nucleotide. It is generally assumed that sugars were formed by Hormuz reaction on the areas. Hormuz reaction is a condensation of small aldehydes, such as formaldehyde and glycolaldehyde. And this reaction can form various sugars, including ribose. And ribose is very unstable sugars, and other sugars must have existed on the prebiotic earth. So it has remained unclear why ribose was selected as a sole nucleic acid component. And previous research found that borate can contribute to the preferential formation of ribose, but it remains unclear whether borate can contribute to the preferential phosphorylation of ribose or not. So we tried the same experiment using another aldopentoses, arabinose, xylose, and rixose. And all of these sugars have similar structure to ribose. We conducted the experiments and compared the yield of each phosphorylated product. Here is the result. The upper left figure is the ribose experiment. The upper right is arabinose, and lower left is xylose, and lower right is rixose. Each yields were 22% in ribose, 8% in arabinose, 5% in xylose, and 11% in rixose. So surprisingly, the ribose phosphate had the highest yield among all aldopentoses in the presence of borate. And here is the result in the absence of borate. The yields were 4% in ribose, 4% in arabinose, 2% in xylose, and 2% in rixose. So there was no apparent difference in the yields without borate. These results show that borate contribute to the preferential formation of ribose phosphate and rixose phosphate than arabinose and xylose. And particularly, ribose phosphate had the highest yield among all aldopentoses. And the difference is probably due to the structure of each sugars. In the present reaction, urea combined with 1' hydroxyl of sugars first, and borate combined with 2' and 3' hydroxyls. And ribose and rixose had the same direction of hydroxyls at 2' and 3' position. So borate can combine with these sugars and these sugars and combine with cyclic form of sugars. And the remaining 5' hydroxyl can selectively react with phosphate. On the other hand, arabinose and xylose have different direction of hydroxyl at 2' and 3' position. And in order to combine with borate, these sugars need to be chain form. Chain form pentoses have 2 hydroxyls at 4' and 5' position, which can react with not only phosphate, but also one more borate. In other words, phosphate compete with borate in this reaction. Therefore, ribose and rixose phosphorylation yields were larger than arabinose and xylose. This means that ribose is the chosen sugar as the point of view of structure, and this might be a reason why ribose was selected as sole nucleic acid component. Previous research found that borate can contribute to the preferential formation of ribose, and we found that borate can also contribute to the preferential phosphorylation of ribose. This finding shows that borate can increase the selectivity of ribose in not only sugar formation, but also phosphorylation. Okay, let me summarize my presentation. This figure shows the reaction pathways of this research and previous research. The upper figure is this study pathway, and the lower figures are previous research pathway. We found that ribose 5' phosphate can be preferentially formed in the presence of borate. This reaction is one-pot, and the yield is significantly high, so we can assume that this reaction is geologically plausible. Previous nucleoside synthesis needs multiple step and complicated procedures. On the areas, one-pot reaction must have preferentially occurred compared to multiple step reaction. These findings open the new route for nucleotide. In this route, phosphorylation occurred first and nucleobase combined or nucleobase construction later. This route is more geologically plausible than previous research pathway, and it also close to the order of nucleotide biosynthesis. Biosynthesis starts from glucose 6' phosphate and converted it to ribose 5' phosphate, and then nucleobase was added to the 1' position and formed the canonical nucleotide. And finally, I'll introduce another research of Borate contribution to the prebiotic chemistry. This is different from the result that we published last year. In addition to the pentose phosphorylation, we are investigating hexose phosphorylation. I'm mainly involved in this project. In this research, we tried the hexose phosphorylation, and Borate also contribute to improve the yield of hexose phosphate. And hexose phosphates were important molecules for the origin life, and particularly glucose phosphate is essential for the biosynthesis of various sugar phosphate. And this is also important in the origin of metabolism. And we'll submit this result soon, so please wait for the publication. And I was not involved in the second part. My boss recently published a new article, which shows that the Borate contribution to the peptide synthesis. This research shows that glycine, simplest amino acid, can be polymerized into longer peptide in the presence of Borate. This shows the synthesis of polypeptide up to 30 nanomolar in the Borate-added sample. And generally, peptide oligomerization proceeds in alkaline condition, highly alkaline condition. But this research shows that glycine oligomerization proceeds in neutral pH in the presence of Borate. This result showed that Borate contribute to amino acid chemistry as well as nucleotide chemistry, and suggested that protein and RNA had been evolved in the same environment, same borate-rich environment. So borate will have played important roles in various reactions of chemical evolution on the areas. In conclusion, we found that borate-rich environment could have contributed to the preferential formation of ribose 5' phosphate. Further chemical evolution for RNA will have occurred in the same environment. So this result indicates that borate-rich environment could have been a probable place for the origin of nucleotide and RNA. This is the summary of my presentation. We investigate the alternative pathway, another pathway of ribonucleotide formation. And the result of the experiment, ribose 5' phosphate was selectively formed from ribose and phosphate in the presence of borate. Surprisingly, the yield of pentose phosphate was highest in ribose compared to other pentoses. So we can assume that phosphorylation pathway is more geologically plausible than nucleoside fast pathway. And in conclusion, borate-rich evaporitic environments will have been a probable place, conducive place for ribonucleotide formation on the areas. All of the results in my today's presentation come from this article that we published last year. And if you have interest, you can download from this QR code. Thank you very much.
Yuta, thank you very much for that wonderful talk. So at this point, we will open the floor to questions for either speaker. If you like, you can ask your question directly, or if you prefer, you can put your question in the chat and I will read them. So would ever like to start? Go ahead. Sukrit, go ahead. I don't know if you can unmute yourself. Oh, James, can you?
There we go.
Yeah.
Thank you very much, James. Thank you to both the speakers for great talks. I wanted to follow up with Yuta briefly. Yuta, you presented some really great work on canonical nucleotide synthesis. Are there any possibilities for using this technique for non-canonical nucleotide synthesis as well?
Yeah. We do not focus on the nuclear-based construction. And so your question is, do I try some other molecules or something like that? Other sugars or
not other sugars, but different nucleobases that are not just the ones that current biology uses, but like things like TNAs or XNAs or something like that might have been present as a predecessor to RNA or DNA?
Yeah, of course, some nucleobases. We consider that it's difficult to form canonical nucleobases, canonical nucleotide. Canonical nucleoside is difficult. Synthesis of canonical nucleoside is difficult compared to phosphorylation. But if some non-canonical nucleobase is easy to form, we can change some pathway. Nucleoside synthesis is fast, and phosphorylation pathway can be more geologically plausible, I think.
Thank you. So I think we'll go with Kevin's question first, and then I will read some questions from the chat.
Hi, two fascinating talks. I have to say, this is really amazing. It's coming from sort of two sides of the sort of the modern prebiotic sort of puzzle, the synthesis, what was going on. And I look at what Nick said and what Yukata said. I mean, it's aptly what Yuta said. If you try a combination of the two, like Nick, your work, if you add borate to some of your ingredients at crucial steps, this could solve the conundrum, I think, because based on my time in the Benner group, when I was leaving the Benner group, a very talented young chemist called Alonso Ricardo took over and carried out the first work with borates and ribose, which showed that borate stabilizes ribose and other sort of 5-membered and 6-membered ring sugars. So this was a really big breakthrough in that respect, and it suggests that sort of complex molecules, sort of prebiotic molecules, occurred in sort of like rather arid regions of the earth, where there was actually a lot where borate tends to naturally form. So I'm just wondering, Nick and Yuta, if you combine things, add a bit of borate to your sort of like synthetic sort of setups, Nick, what would happen? I'd like to see, because this is very interesting, I think we've got these two different, Sutherland on one side, Benner on the other. If we could try and bring them together, sort of like by varying the ingredients somewhat, this could solve this problem. Because I think both of them, in some very different ways, can explain the origins of nucleosides and nucleotides.
Yeah, thanks, Kevin. That's a great point. I'm just trying to recall whether we tried using borate, because of course Benner's work was published when we were looking at these phosphorylations. I can't remember whether we tried borate, but yeah, if we should go back and check, and if we didn't, then we could see if we move in different directions.
I really like your sort of very sort of rather, I suppose, almost obvious, hiding in plain sight explanation for the origin of deoxynucleosides and nucleotides, starting from thiouridine. I mean, it makes a lot of sense, I have to say. The nucleophilicity of the sulfur and the more easily reductibility of a carbon-sulfur bond to give carbon hydrogen. This makes really good sense to me, I have to say. I was very, very intrigued and impressed by this, And this follows in with the paper by Krishnan's paper about it takes two. So was there an RNA world at all? did they all arise together, which is something going back 20 plus years no one thought possible, you know.
Yeah, that's a great point. Ram has a couple of great
Yeah.
papers investigating the properties of hybrid polymers, RNA, DNA polymers, and investigating that idea. And yeah, that fits in beautifully with what we showed here. And I think it kind of makes sense that RNA and DNA are equally plausible. If you look at them from a chemistry perspective, you know, they're of similar complexity. What was unexpected about our work was that we were kind of successful going down the ribo route with the pyrimidines, and then successful with the purines, the deoxyribonucleases. That was an unexpected outcome that fits in with Ram's ideas.
And of course, in the last couple of years, Steve Benner, his group recently published a report of the prebiotic synthesis of RNA triphosphates and the synthesis of RNA oligonucleotides on basaltic glasses and nothing else. So this is another development that happened in very recent years too. So trying to get, so we can see how these things happen, but what was really, and you've shown how our DNA can occur as a result with starting from thiouridine, which was really interesting and very elegant work, I have to say. You know, it was fascinating, and it does explain. It shows a very plausible prebiotic route from ribonucleosides to deoxyribonucleosides, I have to say.
Thank you, Kevin. Yeah, cheers.
So I think what we'll do next is we'll just take the questions in the order that they're given. So the next question is from Sam Lockhart, and I really like questions of this nature because I think it's really important in getting people excited about this field. So Sam says first, thank you for your talks for both or either speakers. Any advice for narrowing down research interests when you were going into grad school? How did organic chemistry or nucleotide synthesis become your specific fields?
It's a good question. Would you like to go first, Yuta, or should I? Okay, well, I can give my perspective. So I was an organic chemist. I was working in synthetic organic chemistry, completely unrelated to the origin of life, for my PhD. And then I did a postdoc in complex alkaloid synthesis. So I guess that you could argue, so I was working in synthetic methods that were unrelated to the origin of life, very, synthetic molecules. Then the postdoc in total synthesis was in biomimetic total synthesis. And that's when I started to become a bit more interested in the relationship between, synthetic chemistry and biosynthesis and the interplay between them. And then you could sort of say I've moved further down the spectrum. But the defining thing for me was when I saw John Sutherland give a talk when I was in Edinburgh doing this postdoc on total synthesis. So he came and gave a talk. I asked him a few questions after the talk, and we got talking about some problems. This was the first time I'd seen really academic work on the origin of life. And and he sort of mentioned that he was looking for people to join his group, and it went that way. So it's a tough question. I think for grad school, I'd probably just look at choosing an environment that works for you. You're confident that you're going to get good training and good support from that environment, because grad school is really just the beginning. You get a good training in how to analyze scientific problems and a scientific workflow. And then, after that training, I think that's when you can really think about whether you want to take a step into something completely different, completely challenging that really interests you. But you can be confident that no matter what you've done in grad school, you've got a background to look at a problem-solving approach for a field that might be completely different. So yeah, I wouldn't stress too much going into grad school. That's my opinion.
Actually, I'm not organic chemist. My major is science, but my supervisor Yoshi Hirakawa give me a chance to study give them a team, the research topic about the origin of life and nucleotide chemistry. So I needed to study organic chemistry by myself, So if you have interest, you don't need to worry about your research field or your major. I think you have interest, you can study a lot by yourself. That's my answer. Sorry, man.
Thank you. So I just want to add to that because I think really interesting question. When I went into grad school, I went in knowing that I wanted to study the origin of life, but there are many facets to the chemical origins of life. I thought that the origin of RNA was the most interesting, so I ended up working for Nick Hudd, who works on this problem as well. I think one of the most important things is just to identify what facet of that problem is the most interesting to you and try and find the researchers who are working on that problem. So I'm going to move on now. A question from AC. Since there are alternative theories of the locality of life's origin, such as evaporative ponds or hydrothermal vents, could the presenters speculate on these different conditions such as UV, temperature, pressure, for the impact into potential synthetic pathways presented? So I think what AC is asking is, to your knowledge, what are the most plausible conditions and localities, and how does that affect your research?
Yeah, the temperature is very important for my experiment because the higher temperature, the sugar is easily broke down. About lower temperature, the phosphorylation doesn't happen. So we need moderate temperature around from 50 to less than 100. This is the most ideal temperature, I think, for the nucleotide synthesis.
Yeah, that's a really a great and always relevant question as well, the conditions that are prevailing. So yeah, for our chemistry, I presented, it's a sequence of steps, right? And not all of those steps are compatible with one another. And this is one of the biggest criticisms of not just our chemistry, but all types of prebiotic chemistry, basically, where, you have these discontinuities in these synthetic steps. And so what we did when we were doing this chemistry was tried as much as possible to have consistency. So one of those things that I showed you was that consistency in the reducing conditions that we invoked. So that's important. Consistency in terms of the UV light exposure. As for what conditions actually did prevail, so that's beyond my field. I'm a synthetic chemist, but I think, you can find, there are general ideas about the general conditions that would have prevailed. But of course, there's also different suggestions that at certain localities or at certain times, completely different conditions would have prevailed. And so I think, as long as you don't invoke things that are drastically different to each other within a sequence, then you can look for a high temperature scenario, which could be plausible in one situation, or if separately you develop a low temperature scenario or a high UV scenario versus a low UV scenario. I think these differences, these subtleties in different microenvironments are perfectly reasonable.
Okay, thank you. So Albert has a question. Albert, if you want, you can unmute, or I could just read it. So Albert asks, he says, thanks for the great talk, very impressive. First question is for Yuta. Following phosphorylation, are your sugars still complexed with borate? He asks because would you need to get rid of the borate afterwards to form the RNA oligomers? And if so, is that done easily, removing the borate?
Thank you very much. The sugars, yes, sugar stay complex with borate after the experiment, but the borate is easily removed from sugars by acidic condition. So we can, if the environment change a little bit acidic, the borate easily removed from ribose. So we can consider that the reaction is proceeded to RNA.
Okay, so continuing on with the chat questions, which we're starting to get a few of them. So Sukrit asks another question for both speakers. We heard today 2 distinct pathways for potentially prebiotic nucleotide synthesis. Is there any way to use the rock record, phylogenetics, or some other empirical tool to discriminate between the two? Yeah, I really like this question because it gets at what evidence is there remaining to judge different hypotheses on prebiotic chemistry. So either presenter, go ahead.
I guess that we cannot find the evidence on the Earth, but perhaps we can find some evidence of chemical evolution on another planet like Mars or some icy moons. If we found some, for example, sugars combining borate or something, it suggests that the phosphorylation fast pathway might be correct. Yeah, but it's difficult to find some geological evidence on the areas because all geological rocks or something sediments were disappeared on the areas due to some geological event.
Yeah, it's a bit of a fantasy, really, to have evidence like that. I think it'd be great. I think we'd all love to find evidence like that. That's a really interesting point from Yuta. Yeah, if we could find it, better preserved evidence on another planet, that would be fantastic. But yeah, I think on our planet, any chance of just, any chemical we find, if it's relevant to the origin of life, it's 4 billion years old, so it's obviously undergone some processing, and B, it's pretty hard to tell it apart from different processes that could be happening on our planet. And when it comes to phylogenetics, phylogenetics can only get you back to a point that has to be after the origin of life, right? So it doesn't really have any bearing. It can be useful to sort of map out general ideas about chemistry and biosynthesis, but modern biology and even ancient biology doesn't really have a strong connection to prebiotic chemistry. They might be the same, they might be completely different. One of those might be necessary for biology to emerge.
All right, so we've got quite a number of questions, which is great. So Albert asks next for Nick. It seems like your synthesis very nicely sidesteps the messy-formose reaction as a source of ribose and deoxyribose. But one of the main attractive features of the formose reaction is its autocatalytic nature. That's interesting. Do you think any of the steps in your synthesis have the potential to be autocatalytic or coupled to an autocatalytic reaction?
Yeah, that's a good point. It's an interesting question. It's sort of like a backhander. You nicely sidestep this messy reaction, but I want to involve this messy reaction anyway. So yeah, our steps, as far as I can think of, because most of our steps are sort of done step by step. So that's not to say, like, many of the sequences we can telescope, and indeed the sort of pinnacle of the work that I showed you, I didn't elaborate on it, was we did as many of these reactions as possible in one pot, and we showed that you could get both those routes. So you could get the route to the pyrimidines that was already existing, and we could convert half of that material in the same pot to the deoxypurines. So those 4 nucleosides existed in the same microenvironment, the same pot in a chemical sense, which was a big advance. But yeah, we haven't really looked at sort of like a continuous development of the reaction, which is where I think autocatalytic networks might emerge. So I'm trying to think if there's any potential. Of course, when it comes to nucleosides, they're complex functional molecules, so I guess there's always potential. But yeah, we sort of look at, we want these reactions to be as clean as possible. That's not to say that we stack the cards too much in our favor. We look at the spontaneous reactivity of these mixtures, but we didn't in this case. Once we got the products we wanted, we didn't sort of let it keep stewing and see which directions they're heading. So I don't know.
So we have a question from Kevin and a question from Alex in 3 parts. I think we'll take those questions and then maybe one more just for the interest of time. So from Kevin, following from AC's question, how likely do you think life is to emerge in the subterranean oceans of icy moons like Europa, Ganymede, and Enceladus?
It's a very difficult question, but I think the possibility is not so high because our reaction is to remove the water from the system, but the icy moon has a lot of amount of water, so it's difficult to remove water from the system. And some reaction was happened in water, like using some reactive phosphate, like triamide phosphate or diamide phosphate or something. But we need to concentrate such molecules to proceed the reaction. But in the icy moon, it's difficult to concentrate or accumulate these molecules in one place. So I think that we cannot expect that life was born in icy moon.
Yeah, I think that's a great answer. I think we'd have to discover some very interesting new mechanisms for accumulation that that defy entropy, really. So it's pretty tough to imagine.
So I'm going to read Alex's 3 messages as one question. So to Yuta, would you expect the composition of the atmosphere to affect the reaction? So the evaporation is occurring under, oxygen here. Does that affect the phosphorylation in any way? How do you know exactly what the reaction mechanism is? What are the possible side effects of dissolved gases such as oxygen or high concentrations of something like bicarbonate, presumably from the presence of carbon dioxide?
I didn't consider about the question before, but basically I do not think the composition doesn't matter for my reaction because my reaction do not need some oxygen or oxidized or reduced reaction, but also CO2 concentration might have some effect because CO2 control the pH of the early ocean. So, for example, highly CO2 atmosphere, the evaporative basin become a little bit acidic and proceed some reaction, I guess.
Yeah, I'm inclined to think the same thing because the borate and the phosphate are both in their highest oxidation states. So yeah, but of course, carbon dioxide can affect the pH. So let's see. So AC asks, question for Nick, rather than UV, could another ionizing radiation such as gamma rays or X-rays serve as the energy source for the photochemical set?
Yeah, great question, AC. So, I mean, we've got Albert Fahrenbach in the audience here. So he's certainly shown that the early chemistry I showed you, the homologation chemistry, can be promoted by gamma radiation, which is thought to be significantly more prevalent on an early, more radioactive Earth. We didn't try our later CS bond scission with anything other than UV light. So yeah, maybe Albert and I can have a chat about trying some of these CS bond reductions with a gamma ray source or similar. Wonderful. What do you think, Albert?
If I may, I would like to ask one last question. I'll take advantage of my position here. So it's kind of in the vein of that previous question. So Nick, when I was watching your presentation, I noted there's a conspicuous similarity between the work you did and a lot of the themes that are going on right now in photoredox catalysis. And like you had EDA complexes And I've seen your other work that has like ferrocyanide as a photoredox catalyst and stuff like that. Do you think there are any prebiotically plausible means of a similar photoredox process that could use visible light?
Great question. Yeah, that would be great if that was possible. So yeah, most of the photoredox now that's happening with blue light or visible light, it's with these fairly complex catalysts that feature metals like iridium and so forth. So I think, yeah, obviously to aim for something as sophisticated as what we see in the literature now is a bit ambitious, but I'm sure there must be processes that, yeah, utilize visible light. I mean, these processes must be spontaneous. Obviously, UV light is higher energy than visible light, so it's able to engage in these photodetachment processes and engage more generally and interact with the bonds of organic matter. Visible light tends usually to just pass through, and unless of course you have more complex chromophoric molecules with extensive conjugated systems. But those molecules I think are plausible. I think it'd be a bit messy. The character, you can imagine prebiotic processes to form those molecules would be polymers and so forth of cyanide and so forth. So it might be, maybe the reason it hasn't been discovered so far is because it's messy, tricky chemistry, but open to discovery, I guess.
Yeah, I like your answer. I remember you mentioned in your talk that there was you called it an encounter complex. To me, that sounds like an electron donor acceptor complex. And you mentioned that it has a color. I think you mentioned it has a color, or maybe I just inferred that.
A color, did you say? Or
yeah, does the encounter complex have a color, a visible color?
No, I don't think I said that. Maybe I misspoke, but yeah, you're right. It's certainly an acceptor donor complex. But yeah, as far as we know, it still undergoes absorption at that at 250 nanometers, 254 nanometers, and yeah, basically just pre-organizes that complex for the donation of the electron into that CS bond, rather than to breaking the pi bond that was happening in the other molecule.
Wonderful. Okay, thank you so much. Thank you both to both speakers, Nick and Yuta. The presentations were amazing. And thank you everyone for attending this session of the PCE3 seminar series. I think we'll conclude with that. And yeah, thanks for coming.
Thanks very much, everyone. Thanks, David. Thanks, Yuta.
Thank you very much.